actually really happy and thankful for Stuart Neufeld. The Center for Evolution and Medicine thanks you for inviting Dr. Michael Eisen here to give our last Center for Evolution and Medicine of the semester series talk. So Dr. Eisen got his bachelor's and PhD at Harvard, then did his postdoc at Stanford, and is now a professor, HHMI investigator in the Department of Molecular and Cellular Biology.
Just cell.
Molecular
and always
and
until
cell.
For some reason it's just molecular and cell biology. No, you'll
Only one cell
I don't know why.
at a time. And in integrative biology, also affiliated with the MVZ, which is the Museum of Vertebrate Zoology. And if you haven't had a chance to visit at Berkeley, it's fantastic. I got to touch a platypus. It was great.
A dead one.
Well, yeah, but it's probably the closest I'll get. So Dr. Eisen, research spans a lot of disciplines, but primarily using computational and experimental approaches to understand the relationship between environmental and genotypic variation on phenotype and phenotypic variation. He's also an advocate for open science and has been really inspirational to me, so probably may not discuss that so much today, but it's worth it for the science too. So let's welcome our speaker, Dr. Michael Eisen.
Thank you. So it's awesome to be here. Thanks for inviting me. I'll just say at the outset, I like interactive seminars. I miss the world when people challenge seminar speakers and ask them questions in the middle, and so feel free to either ask me questions or say that something doesn't make sense or you think is stupid or whatever. So my lab actually has two parts to it. We have, as Melissa said, an interest in the relationship between genome sequence and phenotype. And this has, over the course of 15 years, sort of taken us down, not exactly a rabbit hole, but taking us in kind of a direction that we weren't really thinking about, which was at the outset, which was, I was inspired by all the people who had spent 20 or 30 years arguing for the importance of gene regulation in evolution to when I started my lab, which was really at the beginning of the era of having genomes of animals, to start to try to use these genomes to understand interesting things about how they evolved. But we very quickly realized that this wasn't so easy to do, because unlike studying the evolution of protein coding genes, where we actually know quite a lot about how protein coding information how proteins are encoded in genomes and what the relationship is between a particular DNA sequence and its output, that we really don't have that knowledge in animals. And so we have spent the better part of the last decade trying to understand how animal genomes encode spatial information so that we can, I hope, before I die, learn something about the impact this has had on evolution. In the last couple of years, and I'm not going to talk about this today, but in the last couple of years, my lab has started a bunch of new projects that are, I can't even claim they're related. We happen to do them on the same species, but conceptually they're quite different. But we've been interested in this phenomenon that has been described in a number of different cases where microbes, either bacteria or fungi or parasites, have evolved the ability to manipulate the behavior of animals that they're associated with. And we don't really understand how any of those systems work. So we don't have really enough to talk about from this to fill a seminar. But we started exploring a bunch of interesting systems, all using, for the most part, all using Drosophila or other model organisms rather than going out in the field and taking weird ants and doing it with that. We work on then it turns out there's a bunch of different ways in which model systems are manipulated in the environment, and hopefully maybe in 4 or 5 years I'll be able to come back and tell you something cool about that. Okay, so what I am going to talk about is this question that has intrigued us for a while, which is, we know that animal development is to a large extent driven by differential expression of genes at different stages of development. So sort of anywhere you look in the development of an animal, you find that the creation of novel patterns, the creation of organs, tissues, is preceded by the elaboration of the ultimate spatially segregated pattern in some way by turning on different sets of genes in those tissues. And so, this has been known for decades. But if you sequence an animal genome today, which has become all too easy, if you sequence a new genome of some new animal and you go into the DNA and I ask you to tell me in what cell types or time and development or conditions a particular gene is going to be expressed, you can't do that by just looking at the DNA. You can maybe learn something by neighboring species. You can do lots of experiments to learn it. But we still, 15 years into the era of having the genome sequences of animals, are effectively incapable of looking at their DNA and telling us anything about how particular genes are going to be expressed. And this is a problem for all sorts of reasons. It's a problem intellectually. I consider this to be sort of the greatest unsolved problem in molecular biology, is understanding how genomes encode patterns of gene expression. But it's also a huge problem practically. I mean, I think people who work in medical genetics will know that a lot of disease loci are outside protein coding regions. Presumably, they're in regions of the genome that affect how genes are regulated. But unlike when we can see amino acid substitution in a protein coding gene and have some idea about how to pursue an understanding of what that causes, in the case of gene regulation, we're essentially incapable of doing anything like that because we don't know what that particular region of the genome is encoding. Okay, so we do all of our work in this field in Drosophila and in fruit flies, or I should say, to be pedantic, in vinegar flies. I'm told that that's the proper name, in vinegar flies, and really focused almost exclusively on the first 3 or 4 hours of Drosophila development. And there's a bunch of reasons we work on this system. I mean, there's a long history of genetic dissection of early embryogenesis, so we know a lot of the players that are involved in patterning. It's also, as things go, a relatively simple system. You go from fertilization to this embryo here, which is quite soon after this stage is going to gastrulate, but we don't even get through to studying gastrulation. We study these really 14 cell divisions that happen from fertilization through to the formation of this cellularized embryo. And just to remind those of you who don't remember, Drosophila embryos develop in these initial stages as a syncytium, that is, the nuclei divide without the formation of cells. And this is another reason why this is a good system for studying gene regulation, because unlike invertebrate embryos where you have 2 separate cells from the earliest division, and now in addition to thinking about how genes are regulated, you have all the complexity of cell-cell signaling and things like that to talk about. To a much larger extent, we don't have to worry about that in the early fly embryo. It's governed much more by the generation of transcription factors and their diffusion and so forth. A lot of our work and a lot of the classical work focuses on this one elongated cell cycle that happens about 2.5 hours into development, depending on what temperature you're growing the embryos at. And this is the stage when the very rapid cell cycles of early development come to a pause, and you get an elongated interphase during which the cell membranes actually form. And it's at this point that it becomes much more like a classical embryo with about 6,000 discrete cells, and then it proceeds to start doing all the things that we're used to embryos doing, gastrulating and so forth. And what makes this an important and interesting stage to study this at is that despite the fact that not a lot has gone on morphologically, you've got nuclei dividing, they migrate to the periphery, and then you have the formation of cell membrane. So visually, as you're looking at it under a microscope, it doesn't look that different. You certainly see no visible signs of patterning in the embryo. Underlying this sort of morphological stasis, you have a tremendous diversity of patterns of gene expression. There are something on the order of 1,000 genes that are expressed in clear patterns, spatial patterns, in this cellular blastoderm stage of development. And they're not just random patterns. These are patterns that are laying down the segmented body plan of the embryo, of the embryo and then the larva along the anterior-posterior axis. They're specifying the tissue layers along the dorsal ventral axis. They're determining where the head and the tail essentially are being formed. That is, these patterns of gene expression are laying down the body plan of the animal. And so understanding where they come from is critical for understanding how the fly genome makes a fly. Okay. So, right, I sort of mentioned this. There's several distinct patterning systems that are at play in the embryo. We focus primarily on the anterior-posterior patterning system, in which you have a series of crude gradients of activity that are left behind by the mother in the form of, you can effectively in the form of gradients of activity of transcription factors. And those very crude gradients are transformed into increasingly specific patterns of transcription through the activity of a growing cascade of transcription factors that are themselves expressed in patterns. And I'll come back to talk about that a lot more in the future. There is sort of a parallel and to some extent intersecting system of patterning along the dorsal ventral axis, which has many of the same properties but has different genes involved in different outputs. Okay. So the fact that genes were expressed in patterns was really starting to be worked out in the late 70s and early 80s, but not a lot was understood about how those patterns were materializing. And it really wasn't until and I should say, like, a lot of the way people thought about these things was very heavily influenced by what was known at the time about gene regulation in microbes, especially in bacteria, where you have promoters. The promoters specify where genes are going to be expressed, and they sort of carry all the information for how a gene is going to be regulated. But then a series of papers from Walter Schaffner's lab set out sort of a different path for the study of gene regulation in animals. And what was contained in this paper in 1983, again, I'm biased, but if there's any paper that deserves to win a Nobel Prize that hasn't, to me it's this one. This is the paper that really was one of the key papers in the discovery of a class of sequences that are the main focus of my lab, which are enhancers. And what the Schaffner lab discovered was that there are sequences that are not in promoters themselves, but can be located distal to the promoter of a gene that can influence when and where it is transcribed. Okay. And we now know, because of work that followed on from this initial work from Schaffner and Tanagawa and others, a bit about enhancers. First of all, we know that they can act often at surprisingly large distances to regulate their target genes. So in flies, there are a few enhancers that act over 100 kilobases worth of DNA, which is pretty impressive for a relatively small genome of the fly, although most of them act within, 5 to 10 kilobases of their target genes. In humans, a much bigger genome, there's more real estate. They can act at much larger distances. Again, typically they're within, say, 100 kilobases of the promoter, but there are cases where they are more than 1 million base pairs away from the gene that they regulate. And this is sort of a common property for enhancers, that they act at a distance, and to some extent, it doesn't really matter where they are located near the promoter. It turns out in detail it matters somewhat, but the sort of defining characteristic of an enhancer is a sequence that you can put far away from the promoter that it's regulating, and it will still find the promoter and activate transcription in particular patterns. And of course, there's a lot of complexity underlying that statement, but that I'm not really going to get into, but that's the sort of general idea. It also turns out that there's a lot of them. We don't have perfect assays for enhancer functions, so a lot of this is indirect. But if you use various ways of determining what constitutes an enhancer sequence, we get estimates of maybe 10, as many as 50,000 discrete enhancers in the fly genome, and maybe as many as 300,000 in the human genome. It's maybe slight overestimates, but this is probably the right order of magnitude for them. That is, these are not rare things that work for certain small classes of genes. This is a generic way of regulating transcription, especially during development. And crucially, they're modular in the sense that a given enhancer, it is sort of a self-contained unit. It's an initially defining characteristic of enhancers were that you could take them out of the genome, put them in front of a reporter gene, and they would drive the same pattern in the reporter gene that they would drive for their endogenous gene, and that they act sort of like a kind of a gene regulation exon. Here is sort of the best example of that, which is the even skip gene in Drosophila. Even skipped is a gene that's expressed in the cellular blastoderm stage in 7 rings. Yes?
Let me think of on your invitation.
Sure.
So it seems like you said a sort of a contradiction here, that it doesn't matter where they are, they can act at this distance, and yet if you put them in, you can put them in front of another gene and they act on that gene, but there's got to be some addressing, some specificity to what
Yeah,
you
so
think.
how they find their target promoters is still a mystery. The transgene assays that people tend to do are putting them in a little bit of a rarefied context. That is, when we do a transgene assay, we usually take it out of the genome and put it in a boxed-in kind of construct where it's flanked by insulator sequences. It's nearish to the promoter. The remarkable thing there is not that it finds the promoter, but that it still drives the same pattern. So the pattern is embedded in the enhancer. The ways that enhancers, I'll come to this in a little bit in a second. We have essentially no idea about how in the genome enhancers are actually finding their target promoters. There's a much harder problem. They're solving a much harder problem in their endogenous context, which is they're sitting somewhere, skipping over sometimes dozens of genes to find a target promoter. In these reporter assays, we don't force them to do that job, although increasingly we have the tools to start testing those kind of problems. But yeah, in their endogenous context, they have some ability to seek out and find a particular promoter and regulate its transcription. We in some sense cheat in doing these reporter assays, but the thing that's cool there is not so much that they find the promoter, because we put them right next to the promoter, but that when they're in that context, they recapitulate the same pattern. So I'll give you an example in this case, which is even skipped is this gene that is expressed in these rings around the embryo, these 7 stripes. These are part of what specifies the segmented body plan of the embryo and larva. So they're critical for anterior-posterior patterning. The gene gets its name because its mutants lack even numbered parasegments as the embryo develops. So it's a critical gene for specification along the anterior-posterior axis. If you go back in the literature when this pattern was first observed, people thought this is, you have a repeated pattern, it's going to be some kind of, repressive system where you have adjacent genes repressing each other and you create an oscillator. But that's not at all what happens. In fact, this pattern of 7 stripes, which seems extremely symmetric in the embryo when you look at it, is in fact the product of independent activity of 5 different enhancers. And these sequences, if I take, say, the stripe 2 enhancer here, and I pop it out of the genome and put it in front of a reporter gene, it drives expression in those same nuclei that the second stripe is expressed in the embryo. And the same thing's true for the stripe 1 enhancer, 5 enhancer, 3, 7, and 4, 6 enhancers. That is, some of them encode 1 stripe, some of them encode 2 stripes, but that the overall pattern of expression of the gene is the product of the essentially independent activity of these 5 enhancers. Now again, there's a little, in detail it gets a little more complicated than that, but there's some interaction between the enhancers, but it modulates this activity rather than defines it. It's safe to think about this as the product of the independent activity of these enhancers. And you can, if you chop out a big enough region around stripe 2, you can remove the stripe from the gene, for example. If you chop out this sort of larger region around this minimal enhancer, you lose stripe 2 expression, but you keep the expression of all the other stripes. So it really does behave modularly. And we know something about how those are actually working. So if you take the Eve stripe 2 enhancer, the sequences that matter are binding sites for transcription factors. So earlier on in the system, before Eve comes on, we start with a primary anterior-posterior morphogen that is left in this anterior-posterior gradient by the mother. This is a maternally deposited pattern. It works together with other maternal factors to turn on genes that are canonically called gap genes that are expressed in these very broad patterns. 2 of them here, giant and cruple, are crucial for the regulation of this enhancer. Each of these 4 genes, the anterior activators, bicoid and hunchback, and these gap repressors, giant and cruple, have binding sites in this enhancer sequence. And they work together to produce this pattern. And in fact, this pattern is relatively easy to understand in light of these patterns. Basically, you get activity from this Eve stripe enhancer whenever bicoid and hunchback are present and whenever giant and cruple are not present. So basically, it occurs in a little stripe that lies between this anterior domain of giant and the central domain of cruple. And this is another kind of general property of a lot of these developmental enhancers, that they create novelty because of the intersecting activities of activators and repressors. Again, it can get more complicated, but that's a general principle. So despite everything I just said there, in many ways these sequences are extremely enigmatic. Okay? So we really don't have a good understanding of how they actually work. What is it that they're actually doing to turn on transcription of their target genes? I mean, this came up before. We don't know how they find their target genes. We don't know what specifies what target genes they should have. We don't really understand how their internal, the composition of binding sites that they contain, and this is something I'll come back to, actually specifies their pattern. And as a consequence of that, and in fact another problem, which is really what I'm going to focus on today, is we don't know why some sequences function as enhancers at all. That is, the genome in a particular stage of development, it's not like the whole genome is functioning as enhancers. Only a small fraction of the genome is functioning as enhancers. And that's not because those other regions don't contain binding sites for these transcription factors. So transcription factors bind very promiscuously to naked DNA. So if I take a particular transcription factor, say hunchback, hunchback is one of these anterior activators in the context of Eve stripe 2 at least. It binds basically to a series of Ts. The other anterior activator, bicoid, is TAATCC, and even then it allows for some degree of redundancy in that binding site. Your typical transcription factor binds to about 4 or 5 base pairs worth of information. And even in a small genome like Drosophila, 175 million nucleotides, you find binding sites for each of these transcription factors everywhere. Okay, and this is just sort of an illustration of that, showing you predicted binding sites for this collection of factors that are all involved in anterior-posterior patterning over a megabase worth of DNA. That is, they're just everywhere. And if you take any particular chunk of the genome, it's going to contain a lot of binding sites for transcription factors that are active in that stage of development. So at first glance, you might think that every piece of DNA should function as an enhancer. But we know that that is not the case. I'll show you some data on that in a second. But we know that actually only a small fraction, only a couple % of the genome in the blastoderm is actually acting as an enhancer. And so the question I'm going to talk about today is, why is that the case? Right? Sort of it's essential to understanding how genes function. You need to know how to find genes. I need to know where they are. I need to know what a transcript start site looks like, and so forth. We have good tricks for doing that in the genome. We recognize open reading frames and so forth. So the question is, why doesn't every piece of DNA function as an enhancer? So one model, and in fact the model that we spent the first 5 or so years pursuing, is maybe a kind of obvious one, which is that there are binding sites in these regions, but your instinct when you look at this pattern as a biologist say, well, there must be something that matters about how those binding sites are organized in the enhancer. That is, they're not just a bag of binding sites that you throw down randomly on pieces of DNA, that there's something crucial about how they're organized. And that if I were to take that same collection of binding sites and reorganize them, put them into the same enhancer in a different way, that it wouldn't work. And indeed, experiments that were trying to do this consistently failed. So a lot of people tried to make synthetic enhancers by taking these binding sites and organize them in different ways, and they always failed. Okay. And yet we know that the binding sites really matter. If you take the Eve stripe 2 enhancer and you start to get rid of binding sites, like you get rid of binding sites for the activator bicoid, the pattern goes away. You get rid of binding sites for the repressor giant, the pattern expands into the place where giant is expressed. So it really is the binding sites that are determining the pattern. And yet it somehow didn't seem to be as simple as just throwing binding sites into these regions and you got activity. And so this led to this idea that the reason these sequences function as enhancers is because there's something magical about the way that those binding sites are organized. And this was influenced by early structural studies of some enhancers that showed that they were bound by transcription factors that organized into very rigid kind of arrays. There's the structures of these enhanceosomes that there was this idea that there was something about the way these enhancers are organized that was critical to their activity. However, when people started looking at the evolution of these enhancers, and the pioneering work in this came from Misha Ludwig and Marty Kreitman at the University of Chicago over a series of a decade, they noticed that the positions of these binding sites, including binding sites that were known to be crucial for the activity of the enhancer in melanogaster, were not stable over evolutionary time. And in fact, you saw this great churning where binding sites here, this is a binding site for Krupel. if I knock it out in melanogaster, it affects the activity of the enhancer. But that binding site is only found in this small subgroup of Drosophila species that are all closely related to melanogaster. It's absent from the more distant Drosophila species. And as you see, this is a general rule. Binding sites come and go in these regions. And yet, remarkably, if you take, and this is Misha Ludwig's work, if you take these enhancers from these other species and put them back into melanogaster embryos, they drive the same pattern as the melanogaster enhancer. So that despite this churning of sites, there is a conservation of the activity of these enhancers and a conservation of whatever it is that's reading them out. Okay. So we got very interested in this, and you can perhaps interpret this observation in two almost polar opposite ways. One is you can say that maybe this shows that the structure of the enhancer isn't really critical. And I'll come back to that later. But our initial instinct was, well, this is really great. Evolution is doing a great experiment for us. It's not only conserving the function of these enhancers, it's allowing certain ways of reorganizing the information in these enhancers. We know, because we tried to do it, if we tried to do these experiments ourselves, moving binding sites around, they tended to fail. But if you did them, evolution seemed like it figured out ways of doing them. And so we figured, well, let's just see, it's a classic cheat for evolutionary geneticists to do. Rather than doing the experiment, we'll just take advantage of what the experiment evolution's already done for us. We'll go and look in lots of different species at these enhancers, and maybe we'll learn something critical about how they are functioning. Okay. This is something that happens all the time in evolutionary genetics, of course. We, for a variety of reasons, felt like we needed to look outside the Drosophila to do this. Despite the fact that there's turning over of sites within Drosophila, the overall structure of these enhancers is still largely the same, even amongst the most distantly related Drosophila species. So we did an experiment where we went out further afield into weirder flies, and we ended up working primarily on this group of flies called sepsids that are about 2.5 times as far from melanogaster as the most distantly related Drosophila species is. So we felt that was going to give us sort of more traction on this problem. These are kind of fun flies to work with. They live on animal dung, and we needed fresh animal dung to collect embryos. So my graduate students came up with all sorts of interesting methods to collect fresh cow dung, including running up behind cows and scaring them. And it turns out that they defecate on command. And this is my favorite method section of any of my papers. So this was before it became easy to sequence the full genomes of species. So we went in and pulled out the regions of the genomes of these flies that corresponded to the even skip gene and several other loci in melanogaster. And with a lot of painful work that I won't describe, we were able to isolate enhancers from those species that corresponded to the melanogaster enhancers. And what we found there was that process we started to see underway in Drosophila just kept on going, that the enhancers in these other fly species were effectively completely reorganized. There's one little cluster of binding sites that maybe is conserved, but the rest of it's completely reorganized relative to melanogaster. And yet, if I take those enhancers and pop them into a Drosophila melanogaster embryo, it drives a pattern that is indistinguishable from the pattern driven by the melanogaster enhancer. Okay. So again, there's two ways you can look at this. You can look at this as saying, well, there's not really any necessary pattern. There's no required thing here. But we thought, well, maybe there's some pattern there that we were missing. So we spent quite a lot of time analyzing a large collection of these enhancers from sepsids and other species, and we started to notice a pattern. And the pattern we really started to notice was that the binding sites didn't look randomly organized. They tended to be organized into small blocks of sites, and that especially when you saw small blocks of sites together, those sites tended to be highly conserved. So we really thought we were onto something, that these enhancers were organized in a modular manner, and that there was some kind of internal structure to these enhancers that was critical for their activity. Okay, so as that work was happening, this project has sequenced a dozen Drosophila genomes, reached maturation. We now had a lot of data from Drosophila to go back to. And we figured, well, let's see if this pattern continues across all the Drosophila species. But before we did that, we wanted to make sure that we weren't being tricked by, we had a good model for understanding what we were looking at. And so a graduate student in the lab said, well, let's just simulate this process we're looking at, right? The process as we understand it is you don't, that binding sites can come and go, not because they're not essential, but because the small size of binding sites makes it very easy for evolution to create new ones. So random mutation will create new binding sites for these factors quite frequently. And just like gene duplication, we figure that when new sites are created, if they're in the right place, in the right context, they create redundancy, and the original site can be lost because of the new site. So the first thing we wanted to do was say, okay, what's the null model here for our comparison? And the null model is that that binding site turnover happens without any constraint on where the site should be found. So, right, we have a hypothesis that it matters how the sites are organized relative to each other. So our null model isn't that the sites are static, but that they turn over, but without any constraint on where they're going to be found in enhancers. And so we did this. We actually, we wrote a simulation of this in which basically we created sort of synthetic enhancers and embedded them into real Drosophila DNA. And this, again, is entirely in a computer. And we created an enhancer that needed to have 5 bicoid sites and 5 Krupel sites. And then we just let them evolve. We let them evolve by drawing random mutations from what we know about the Drosophila mutation spectrum, both the substitution frequencies and insertions and deletions, and then just let it go. And basically, if a binding site was destroyed by mutation, dropping the required number of bicoid sites in this case below the necessary threshold of 5, that mutation was not accepted. It was viewed as unfit. But if a new site was created first, then another site could be lost without consequence. And so you could get binding site turnover. And this is what the results looked like. And I don't know, do you notice anything? So let me just say what this is. These are generations in our simulation. These are the binding sites distributed across the enhancer, and this is just time. Okay? So you can see sites coming and going, right? Here's a site that was present at the beginning and is lost because a new site came up over here and so forth. So you see some churning. But you'll probably also see another pattern. Does anyone see another pattern? What's that?
Parallel?
Well, they're not exactly parallel, right? Like if you look at it, they sort of are, the lines, especially the longer ones, are sort of moving inward, right? And it turns out that there's a phenomena that takes place in essentially all animal genomes that we've looked at, which is that insertions and deletions are not symmetric, right? You have a strong bias at the kinds of short changes that we were looking at in these simulations in favor of deletions over insertions. And then you have a bias in favor of insertions over deletions for large things, transposon insertions and things like that. But we know from observation that transposons tend not to insert themselves in the middle of enhancers. And so we actually found, yeah?
Is that because selection, or?
We don't know. We just know that in all non-coding DNA we've looked at in Drosophila, there's a bias for deletions over insertions, whether it's selection or not. It could be selection, and we assume it's not. We assume that it's just a neutral mutation bias. But this is true in humans, that there's this bias, and at any size scale you look at, there's not an equal number of insertions versus deletions.
You said there's a bias against the transposon.
Yeah, sorry. We think that's selection. We think that if you insert a transposon in an enhancer, the enhancer won't work anymore. Right, so, sorry. Yeah, sorry, that's right. That's not a mutation bias, that's a selection bias. But it meant that actually our simulations, without us even realizing it, had this property that if you have 2 sites and they stick around for a long time, just by chance, that is they're not lost or turned over, that they will get closer to each other, because you'll be deleting more sequences between them. And that this creates an entirely artificial relationship between conservation and clustering. And that in fact, you can create a genome in which important binding sites are all clustered together without there being any selection for that phenomena. Okay, and we went back and analyzed this and thought about this a lot, and it turned out that as best as we could tell, all of the structure of enhancers that we were observing in Drosophila could be explained by this kind of neutral model. Okay. And so because of this, and actually, so we sort of coined this idea that this clustering of structure of binding sites is actually kind of a mirage. It's a mirage created by this deletion bias. So because of this and a bunch of other reasons, we began to really become skeptical of the idea that what it was that was making enhancers function like enhancers was the way binding sites were organized within them. Okay? But it still raises the question of why are they functioning as enhancers, right? Why is it that such a small fraction of the genome functions as enhancers? And over the same period of time we were doing this, a kind of another way of thinking about this was emerging from studies of histones and nucleosomes across DNA. And I think anybody who does anything with eukaryotic genomes will know that the genome is not just sort of one uniform string. Obviously, it's got lots of histones, and those histones are not distributed uniformly across DNA. First of all, there are regions of high and low nucleosome density. And then the nucleosomes themselves are not uniform. They're modified in various different ways. And the genome in any particular cell type or stage of development is very highly differentiated. There are regions that are that travel under the name of open chromatin, or what exactly that means. But phenomenologically, what it means is that there are regions of the genome that are accessible to being bound by transcription factors, and regions that aren't. Okay? And you can actually explain, we had a paper looking at this, you can pretty much explain the genome-wide binding of transcription factors by just saying, well, there are regions of open chromatin. We can define those experimentally. There are regions of open chromatin, and transcription factors will bind to their binding sites if they're in regions of open chromatin, but they won't bind to their binding sites if they're outside of those regions. And so great. So we have this alternative idea, which is that the reason that enhancers are enhancers is because they're functionally because they're sort of different chromatin state. But it doesn't tell us why they got to be that way in the first place. So, okay, so we started to learn something about that, really because of kind of a chance observation. And the chance observation was this, that to study, in parallel to all the evolutionary work we were doing, we were collaborating with a biochemist at Lawrence Berkeley National Lab, Mark Biggin, to look at the genome-wide distribution of transcription factor binding in these cellular blastoderm embryos. And the experiment is simple. You probably all know it. You take a large collection of embryos, you dunk them in formaldehyde. The formaldehyde cross-links proteins to DNA. We purify chromatin. We shear it up into little pieces. And then we come in with an antibody to whatever transcription factor we're interested in. We pull out the regions of the genome to which it's bound and then read them out. Initially, this was done on a microarray. Now we do this by sequencing. But the end result is that we get a genome-wide map of the binding of these different transcription factors. And there's a lot to be learned from this and so forth. But when we first started doing these experiments, we were nervous that you're working with antibodies. Antibodies are finicky reagents. We weren't sure that the regions we were pulling down were really bound by that transcription factor. The antibodies cross-react, even though we'd taken a lot of care to prevent that from happening. We spent a lot of time doing these experiments. We spent a little bit of time just looking at the data to try to make sure they were believable. And one of the things we always looked at was to say, okay, here are the regions bound by bicoid. I look in those set of, say, 1,000 sequences that are bound strongly by bicoid, and I ask, are of the sequences that are enriched in those regions relative to the rest of non-coding DNA, do we find the bicoid binding site? Okay, so the analysis here is really trivial. I like to point out this was the last thing that I actually did myself in the lab. So this was just taking not entirely true, but it's the last thing that ended up in a paper. You just take the regions that are bound by this sequence, 1,000 base pairs or so, and then just count up the number of each of the 7 base pair words that are found in those regions, compare it to a background collection of unbound non-coding DNA, and then just ask, these are the top 10 most highly enriched words in the regions bound by bicoid, and comfortingly, we find that a bunch of them correspond to the known binding specificity of bicoid. So we would consider that to have been a successful experiment. We do that for Krupel. Again, you get a different collection of motifs, and amongst them are the known Krupel binding sites. You do that for Hunchback, you get the Hunchback motifs. Okay. But something else started to pop up. Okay. Every time I did this analysis, another sequence always came to the top. Okay? Doesn't matter what factor we were looking at. The same 7 base pair sequence was always at the top of the list. And I'm worked with enough genomes, so my first instinct was to say, okay, this is some bizarre artifact, there's some repetitive element that's doing this, but indeed that's not the case. There are in essentially every, it's molecular biology, so every means 80%. So in essentially every enhancer that we looked at, there was this 7-base pair motif, usually multiple copies of it. And it just happened to be that motif had been noticed by Tom Klein's lab, who was just down the hill from us, who had been studying the genes that are turned on in very early Drosophila genes. These are the first genes that are basically the first genes that are transcribed in the embryo around mitotic cycle 7 or 8. These happen to be involved in sex determination, but he had been studying these genes and what was responsible for their early transcription for many years. And initially a postdoc and then a grad student in his lab got focused on the fact that there, in all of these genes they look at, and by all, this was really like 5 or 6 at the time, they noticed clusters of this motif, this CAGGTAG motif. Once we saw that this sequence was found in enhancers, we got very interested in figuring out what it was. Primarily because of Tom's early work, and I think at least partially because of our observation, Chris Rushlow's lab at NYU went out and did a very simple experiment, a sort of one hybrid experiment, taking that motif and expression constructs from E. coli, and they identified a protein that bound to that motif. So the protein she identified she called Zelda, after the video game character. She has some elaborate excuse for why it's called Zelda, but it's really after the video game character. And it has the property, first of all, that binds to the sequence. It had actually been previously identified, mutations in this gene had been previously identified as having a severe effect on early cell division. So it had already been identified as an important gene in fly development. And by all rights, we should be calling it Vielfältig, because that was, Drosophila geneticists tend to be very picky about naming, and it should have been called Vielfältig, but it isn't. It's harder to say. Okay, so Zelda is a CAGGTAG binding protein. It has some very interesting properties. It has a zinc finger cluster where that binds to DNA. The rest of the protein is just, pretty much the rest of it is a mess of repetitive sequences of various forms. You pick your favorite amino acid, there's probably a long polymer of it somewhere in Zelda. There's polyglutamine, polyalanine, polythreonine, polyserine, polyproline, and so forth. It's your classic, almost transcription factor on steroids with these disordered domains up the wazoo. There are a few highly conserved domains. I should say it's only found in arthropods. There are a few highly conserved domains that we have no idea what they do. They're not homologous to anything else. There's another zinc finger that really is of unknown function. So it's an enigmatic protein. It's proven biochemically to be almost impossible to work with because of all this repetitive stuff. We have a little bit of a handle on it, but. The effects of mutations in Zelda are severe. So this on the left is a movie of normal Drosophila development where you see the cellularization happening, you see gastrulation and so forth. You don't see any of those things in Zelda mutants. The most common phenotype we see for these Zelda minus embryos is that they try to gastrulate before they've cellularized the embryo. And as you might imagine, that doesn't end well. Okay, so we knew from a lot of our binding work, we already knew that some patterns were emerging from this binding work, one of which was that all these enhancers that had been previously identified turn out to be real hotspots for transcription factor binding. Okay? And so we got interested in the idea that maybe Zelda was playing a role in defining where these hotspots are. So the first experiment we did was to go into embryos. We were really good at this point at doing chip experiments where we pulled down things with antibodies. We got an antibody. We actually worked with a woman in Tom Klein's lab and Mike Botchin's lab who was interested in Zelda. Who had made an antibody for Zelda. And we did the chip experiment. And what we found is that in these cycle 14 embryos, Zelda is bound to essentially every enhancer sequence. But it turns out that's not that interesting an observation, because it turns out that basically that's the only place anything binds in the embryo, and everything binds to those sequences. So, but we knew something about Zelda that differentiated it from these other factors, which is we knew from Tom's work that those CAGGTAG binding sites were critical for the activation of these early transcribed genes during during fly development. And we got interested in the question of whether Zelda might be doing something at these enhancer sequences. Right? I should say that in the cycle 14 embryo, it's bound to all these enhancer sequences as well as to the promoters of basically every pattern gene in the embryo. Okay, so it's consistent also with Tom's observation that it was critical for activating these genes early in development. But so we got interested in the question of whether Zelda might be doing something very early in development to impact the maturation of enhancers. Okay, so in principle, this should have been a very easy thing for us to investigate by just doing the same kind of chip experiments we did in later development, but doing them in earlier embryos. But here, flies turned out to have a little bit of a problem for us, which is, right, the way we do these experiments, we have a big cage filled with flies, and we keep those cages going because we like to collect lots of embryos. When we need embryos, we shove a plate in covered with yeast, which the flies like to lay eggs on. And then we leave it in for half an hour, then we pull it out, and we put it aside for however long we need it. We want to collect embryos that are 3 to 3.5 hours old. We collect for half an hour and then put it aside for 3 hours. If we want to collect embryos that are 90 minutes younger than that, we should just be able to not age it as long. Right, just let it go for 90 minutes. But it turns out that fly females are not entirely cooperative with this experiment. And one of the things that they do that's kind of annoying is they don't immediately lay their eggs after fertilization. So in any collection of embryos that you get, you have mostly the stages you want. This is if we're doing a collection targeting mitotic cycles 8 to 10. About 90% of the embryos are in those stages. But maybe 10% of them are older. And again, most of the time in molecular biology, you're like, 10%, what's the big deal? But it turns out that this is a particularly big problem early in development because these older embryos have a lot more nuclei and a lot more DNA. So somewhat paradoxically, if you're doing a collection that's targeted to mitotic cycles 8 through 10, the vast majority of your DNA is actually from older embryos. So this wasn't good. We thought about clever ways of getting around this and tricking the females into laying their eggs at the right time and so forth, but eventually we settled on brute force, which was to sit in front of a microscope, and again, not me, people in the lab, sit in front of a microscope and just sort all these embryos by hand. Every single embryo we look at, and I'm talking about 20,000 embryos for these experiments, is looked at by hand and sorted. So we target a stage, and basically it's not quite as bad as it sounds. You look at a field and you get rid of any embryo that's not the right stage. But this took about 6 months of sorting to do this one experiment. We've gotten better at it, but this is a lot of work.
How many people for 6 months?
2.
2 people?
Yeah, they didn't only do this for those 6 months, but probably it was about 4 months worth of human labor time sorting embryos for this one experiment. So and these were a postdoc at a highly paid tech. It wasn't we didn't train undergraduates to do it at this point. So, but anyway, we did this. We got very, very clean pools of embryos on which we could do these experiments, and that allowed us to look at Zelda binding in the early embryo. And the observation there was just as striking, which is that in this much earlier stage of development, mitotic cycles 8 to 10 was when these embryos came from. Now I should point out that most of the genes involved in patterning, most of the things that are bound to these enhancers I've talked about, they haven't even been transcribed yet. Okay? So if you have a model that requires all these transcription factors to be around to decide where enhancers are going to be, it can't be active at this stage of development because all those factors aren't even present. A few of them are, the maternally deposited ones are. And again, Zelda is, I should say, is maternally deposited factor. But it's bound to all those same places. It's bound to the enhancers. It's bound to the promoters of these genes, even though the vast majority of these genes aren't being transcribed yet and the enhancers aren't doing anything yet. We'd done a bunch of other experiments to do using single embryo RNA sequencing to get a very precise picture of transcription. And you saw one thing we observed was that the stronger the Zelda binding in the early embryo, the earlier the gene is going to be transcribed. But that really wasn't our primary interest. We were interested in what was going on at enhancers. And it turns out that at enhancers, we see not just in looking at Eve, but consistently across the genome. And here's a picture I'm just showing you. Eve, each of these lines represents one enhancer. The enhancer is in the middle, and we're showing you 6 kilobases on either side. And then the color shows you the strength of Zelda binding. So red is the strongest binding. And you can see pretty clearly that Zelda binding is concentrated at these enhancer sequences. And indeed, Zelda binding precedes the binding of basically every transcription factor we've looked at. That is, the enhancers that are bound by bicoid, caudal, and again, these are overlapping sets, so these are not entirely independent observations. But the factors that are bound, the enhancers that are going to be bound by all of these factors, including many that are entirely zygotic factors that aren't even present at this stage of development, are bound early by Zelda. And Zelda, this observation, at least on the surface of it, starts to answer this question that was plaguing us for such a long time, which is, why is it that these particular regions of the genome seem to be functioning as enhancers when other sequences don't? And just to put some numbers on that, if you take bicoid, its binding specificity is basically TAATCC. You look at all the bicoid binding sites in non-coding DNA in the genome, only about 2% of them are actually bound by bicoid. 98% of them are not bound by bicoid at all. If we do a very simple analysis and say, let's just consider only places where there is a bicoid binding site near to a place where Zelda is bound, now all of a sudden 60% of those are bound by bicoid. That is, going from 2% to 60% is a huge, huge, huge difference. Okay? And that's true for every factor we look at. That it binds to a tiny fraction of its sites, but if we say, well, how about the sites that are bound early by Zelda? That number shoots through the roof. And indeed, we don't even have to use Zelda binding to do this, because Zelda, in non-coding DNA, binds to a huge fraction of its own target sites. Very unusual for these factors. And maybe this is because of the stage at which it's active, or maybe it's something specific to Zelda. It binds to about 70% of its target sites. At least its very high affinity target sites. 70% of them are bound by bicoid, and are bound by Zelda. And using that information alone, we can go from specificity of about 2% for a bicoid binding site to over 50% for a bicoid binding site that's near Zelda. So I realize I've taken longer than usual, so I'm just going to go through one little model here at the end for what we think is going on. Okay, so this obviously raises the question of what is it doing? Okay, and our way of thinking about it is that something happens between the cycle 10, when Zelda is bound to these regions, but probably not a lot else is going on on the genome, to cycle 14, when these Zelda-bound regions are now biochemically distinct from the rest of the genome. They have different histone marks, they have lower histone levels, they're more accessible. I'll go through this quickly. This is just some data that shows that these Zelda-bound regions, these regions that are bound early in the embryo, are really very strongly destined to be regions of open chromatin later in development, and that association goes away when we take Zelda out of the embryo. We take maternal Zelda out of the embryo. And basically, we think, and I'll just flash through some data right at the end that supports this idea, that Zelda is triggering a series of events that are happening at DNA. That is, if you have no Zelda site in your DNA, there is a default pathway that involves polycomb and all these other systems that are active in modulating chromatin in the embryo. There is a default pathway that leads to those regions ending up as inaccessible chromatin. They're closed chromatin with all of its associated chromatin marks and properties, and most crucially, it's difficult or impossible for transcription factors to bind to these regions. Whereas if you have Zelda binding site, you're bound by Zelda, that this alters the fate of these regions in some way that leaves them accessible at the end. And I'll just point out that this is now starting to sound a lot like this class of transcription factors called pioneer transcription factors that have a related, although distinct, characteristic that they can bind to closed chromatin and open it up. But they both Zelda and these pioneer factors have the same effect. That is, they are the things that are responsible for determining where other factors can bind, not by recruiting those factors directly themselves, but indirectly by affecting the state of chromatin. And to further look at this, we did another crazy experiment that involved a lot more embryo sorting, although we were better at it at this point, in which we looked at histone marks. I'm not going to go through all the data, but trust me, it's been published, so you don't have to just entirely trust me. But I'll say one thing which is important, which is that these data, and we had a trick in the way we did these experiments, in which when we made chromatin, we added, this is Drosophila melanogaster, sorted Drosophila melanogaster chromatin. We added a small amount of chromatin from another species, Drosophila pseudoobscura, which allowed us to be able to look quantitatively at changes in binding, not just qualitatively. That is, we could use this because we used exactly the same sample of pseudo-obscure chromatin, and all these antihistone antibodies work just as well on Drosophila pseudo-obscura as they do on melanogaster. So we were able to make quantitative comparisons. And the crucial result of the quantitative comparisons was that the early embryo is not, the chromatin is much less interesting. There's essentially no histone methylation. There's a little bit of histone acetylation, but it's much lower levels than there is in the mature embryo. And that, again, this is just the data on where those marks are distributed. I'll show you what the end result is. That basically these data point to the following model, in which Zelda sites are present early. Zelda protein reaches its peak levels at around mitotic cycle 7 or 8. Zelda RNA is deposited by the mother, and then it's translated by the embryo. So if you look at the Zelda concentration in the nucleus as a function of time and development, it peaks around mitotic cycle 7 or 8. So Zelda binds to these regions. We know that's true experimentally. This histone data suggests that early Zelda binding, either directly or indirectly, leads to the very specific acetylation of those regions of the genome that are destined to be active enhancers and promoters later on during embryogenesis. And meaning that before they are active, before they're active as enhancers, before the other transcription factors are coming along, these regions are biochemically distinct. And then that early histone acetylation is followed by distinct patterns of histone methylation that lead to these regions being quite distinct by the time you get to the end of cycle 14. Okay? And this all put together leads to a model that, and this is my last slide, so comes to get a model which is that we think that, in contrast to what we thought at the beginning, where you have a bunch of patterning factors, the patterning factors are binding to an enhancer, and the patterning factors have to do two things. They have to both say, this is a region of the genome that's going to function as an enhancer, and it's going to drive this very precise pattern expressing just in these nuclei of stripe 2 or stripe 3 or 7 or whatever. That instead, there are two separate systems at work here. There is one system of which Zelda is a part. We think there's other players. There's STAT protein, there's a GAGA protein, there's other things we think are involved in this system that don't contain patterning information themselves. What they are doing is they are patterning the genome and saying, here are regions of activity, here are regions of inactivity. They are greatly reducing the complexity of the problem that has to be solved by the patterning factors themselves, and allowing the patterning factors to basically function at what they're good at, which is determining patterning output. And I think that this model, I mean, I would say we haven't fully shown that this is the case yet. There's still a lot of pieces to fall here. But we're pretty confident that this is what's going on in the early fly embryo. And there's reasons to believe that similar things are happening in other systems. There's some preliminary data from other labs in zebrafish suggesting that PAL5F1, which is the OCT4 homolog in zebrafish, is maybe playing a similar role during early zebrafish development. The logic of this, right, matches up with the logic of pioneer factors, that it seems to make sense that you would want, or that it would be easier to evolve systems like enhancers if you don't have this same job taken up by the patterning factors have to do everything. That you would have these these spatially uniform factors that are determining where regions are active, and the patterning factors themselves are just doing patterning. And so this is the model we're currently operating under. We're trying to refine and elaborate it, identifying other components that are involved, and really trying to figure out exactly whether, to what extent is this true universally across fly development. We're starting to look in other species to see how much this is held. And with that, I will stop. The people in my lab who were responsible, so I covered quite a long span of research here, starting with some very early work from graduate students, Ben Berman, Alan Moses, and Derek Chang, Dan Pollard, who were looking at the sort of complexity of the problem of finding enhancers in the early embryo and how binding sites are evolving. Those dung fly experiments were done by two awesome graduate students, Emily Hare and Brant Peterson. The simulation work was done by another great graduate student, Rich Lusk. The Zelda work I've talked about is wow, I left some people off, so I'm going to have to Oh no, they're up higher, sorry. The Zelda work was primarily the efforts of Melissa Harrison, who was this postdoc who was officially in Mike Botchin and Tom Klein's lab, but ended up working mostly with us on this Zelda story. Xiaoyang Li, who did, along with Melissa, did almost all the embryo sorting. And I left off Tommy Kaplan, who was the computational guy who was really tied all this stuff together. And with that, I will stop and happy to take any questions. So thanks. Oh, you got the mic.
Time for one or two questions only.
Sorry.
So are Zelda sites alone sufficient to overcome any heterochromatic region? And with overexpression or promiscuous expression, do you see any sort of position effect?
Yeah, so the first answer is we don't exactly know yet. We've done a lot of experiments with Zelda, taking Zelda sites in and out of enhancers. But those were all done in these transgene assays where we realized sort of after doing those experiments that there's a problem with the way those experiments are set up, which is that they're flanked by the flanking of these sequences with insulators prevents, even in inactive enhancers, so even when we have enhancers that aren't functioning, they're not forming closed chromatin. So we think that this default pathway doesn't actually operate on we've selected regions of the genome and constructs that are immune to the normal silencing that happens in most of the genome. So we are now going back using CRISPR to edit endogenous parts of the genome that we know are in closed chromatin to insert Zelda sites into them and see what happens. We don't have the results yet, but that's the right experiment to do. It turns out that yeah, I mean, there's a lot, now in retrospect, I think a lot of the experiments that were done in these transgenes are problematic because of the entirely, not just the artificial nature of the constructs, but because they're targeted to regions of the genome that have unusual properties that happen to make them good for doing reporter assays, but bad for learning anything about how enhancers are functioning. So,
I'm afraid our time is up. Thanks very much, everyone.
thanks. Sorry for going a little over. But